We are very proud that you come, and if everyone would know you, they would immediately do the connection to AmBisome and to Remdesivir. And we are very interested and happy if you tell us about the trials in nanomedicine that you have been doing and telling us about the paradigm which is in the field of liposomes. I think that is very important, and you are as an executive director in technical operations, the real best person we could invite to speak about this.
Well, it's my great pleasure to be here. Thank you so very much. I wish I could be there physically, of course, but I'm coming to you from early in the morning in Southern California. So I want to talk a little bit about what really was an old but still thriving precision nanomedicine, AmBisome, and that came from the NeXstar company many years ago, and now Gilead, liposomal paradigm. And then I'll talk in the second part of the talk a bit about the Remdesivir product, which is a new Gilead injectable for COVID. So AmBisome is on the market in Europe now, in some countries for 30 years. So it's one of the very first repurposed generic drugs that had been placed into a nanocarrier system, in this case, a liposome system, and really found a new birth, continuing really growth in clinical application. It's approved in many countries around the world, 63 countries at this time. So it came from a classical, what we call a conventional liposome construction. Stable conventional liposomes, they're called, sub 100 nanometer particle size. They focus on long chain saturated phosphatidylcholines and cholesterol. And they emphasize in design and manufacturing vesicle stability. The original products that came out of the NeXstar team were oncology based actually, and looked at some of the tumor targeting ideas that you'll see elsewhere in the meeting, and of course are well known with these small nano systems. In this case though, some of that structure of reticular endothelial system avoidance was enabled, but also some RES uptake was encouraged, or macrophage uptake, by the inclusion of a negative phospholipid in DSPG. So looking at the product itself as a pharmaceutical entity, it has a 4-year shelf life at 25 degrees, which is pretty respectable. I think we're up to 63 countries now. In most countries, it has a very broad label, which is fever of unknown origin. And this essentially is a way to say that for a fungal infection, often the pathogen isn't known, but it's presumed to be not viral or not bacterial. And one of the things I'll try to show you today is that learning the disease is as important in this case as developing the drug itself. It's approved also as a refractory to the old free drug, HIV-related cryptococcal meningitis, a parasite known as Kala-Azar, which I'll talk about as well. And the lipid system substantially reduced toxicity. This is really the main result of the formulation, although there are some targeting elements, which I'll show you as well. And whenever you have lower toxicity, often the clinicians can dose at higher levels. The drug itself is, in the liposome world, perhaps kind of a unicorn. There's really only one drug as well suited for lipid delivery as this one. That's why you don't see a lot of similar products of the same design. Amphotericin B was discovered in the 1950s. It's a polyene. These are 3 versions of the structure. The first 2 are the single species. It is thought and fairly well demonstrated that it functions as an ion channel in either 8 or 16 members. And this conducts monovalent ions to escape from the cellular entity that's been targeted by the drug. And in the case of a fungal particle, that leads to cell death. This can happen with mammalian tissue or with fungal cell systems or yeast cell systems. The big difference being, though, that ergosterol, which is the sterol in those fungal and yeast systems, is of a tenfold higher binding affinity, and that really allows for a very stable carrier to circulate for a long time and deposit the drug only really practically when the pathogen is encountered. So again, this is a passive targeting, but a very effective one. And so these are the two sterols. They're just very similar, the one being the primary one in mammalian membranes, the other in yeast, fungi, and we'll find out later as well, the Cala-Azar parasite. This is a kind of a demonstration of that drug retention. So you see the circles are the AmBisome formulation, and the squares are the free drug, which is really a detergent suspension. And as you incubate with higher and higher levels of drug, in the case of the free drug, the drug partitions very quickly. In the case of the lipid binding systems, which are some alternate ones are the diamonds and triangles, AmBisome is the circles, you get practically no distribution of drug during this incubation from that carrier vehicle. And there's no thermodynamic preference for the drug to depart that carrier and get into that cell membrane. If I do the same experiment with yeast cells as a substitute, all of the curves coalesce. And this tells you that the binding affinity difference of the sterols dominates the physics against all formulations. So it's kind of a in vitro demonstration really of therapeutic index, if you will, that's presented. This translates into lethal dose. This is mirroring lethal dose. You can see the conventional drug at around 1 to 2 milligram per kilogram. And the AmBisome product is up at 175 milligram per kilogram. So again, a substantial opportunity to dose higher or to dose more safely. So originally the label was focused on studies against conventional drug, against another lipid system, and focused mainly on those safety benefits in infusion-related reactions and kidney toxicity. And these studies are really now quite old and they've powered the product's growth over these years. But the company continued to pursue the agent as it started to understand its mechanism of action better. And it did seem that it was likely the drug would target the lung in an efficient way. And the drug itself also has a very broad spectrum of activity against really all active pathogenic fungal and yeast species because of its simple, in essence, its simple mechanism of action. If there's ergosterol present, typically the drug does quite well. So studies were done around the 2007 timeframe to look at Aspergillosis, which is a mostly fatal lung infection. It was carried out again. Usually you don't have the specific identity of the organism in mind. And it was against proven and probable mold infections, usually with the fever and typically with some immunocompromised status. The comparator of interest was voriconazole and the conventional free drug. And there was a lot of in vitro work that went upstream into this. And there were 2 dose levels chosen. And the product performed very well at a relatively intermediate dose of 3 milligram per kilogram per day. The study was presumably dominated by Aspergillus, but as we move forward in time, we realized that usually the lung infections aren't typed as to species at the time of treatment. And so the AmBisome product has done well in this market because of the typical uncertainty and the lethality of the infections. And the key thing is really to know when to dose and what to dose with in the system. This is the result of the 2 dose levels. And again, we're looking at better than 50% survival in a very, very sick patient population. I want to look at some just general ideas around fungal infection. And I borrow these from Professor Ben DuPaul, who kind of has made some excellent illustrations of how the disease works and how therapy fits into it. First, you can't get rid of the pathogen. The world would not be a happy place if there were not fungi or yeast, not least among which for some of these favorite things we enjoy. And so you really can't rid our environment. Some diseases you can imagine the eradication of the pathogen. In the case of yeast and fungi, you simply can't. It cleanses nature, it's essential, and we have to learn how to live with it. So how do we live together with these pathogens? That's the question. If you're a treating physician in a hospital, you really are fighting this terrible prevalence and commonality of these elements. No different than we do in pharmaceutical clean rooms. We try to keep these species out, and it's nearly impossible to do. So you're always at risk. An example of the defense systems being damaged, this is kind of a illustration of a chemotherapy or radiochemotherapy session and looking at the decay in the various elements of the immune system as you go. And there's risk for bacteria, especially early, risk for viral infections, and risk for fungal infections that span the various timeframes. And you can see fungal infection risk is prevalent nearly through the entire duration of the system. And this patient could take many days before they're really out of jeopardy and their immune system has reconstructed if it's going to. So this is an example of when you dose versus mortality, and this was a candidemia study, again showing that if you dose quickly, you have a great opportunity for treatment, and if you wait even a day or 2 days, a much larger fraction of your patients will pass away. And again, this is common. This is just one example. So your odds of controlling the disease decrease rapidly as the evolution of the disease itself continues to grow. And again, if you think about some of these Aspergillosis elements, they can grow to the order of many centimeters in the body over the course of just a day. So you really have to get in front of them. So what it really means, though, is in many cases, you're either trying something prophylactically, or you're going with those empirical signs of early disease, or you're really focusing on waiting until you have a firm diagnosis, including the pathogen, so you can pick the correct chemical agent. In each case, and this is an example for Aspergillosis, you're dosing a large number of patients who may or may not have the actual element. And so this is something that physicians have to continue to work towards. We continue to study mechanism of action, even though the product has been around for a long time. Many of its favorable properties were sort of mysterious in terms of how well they worked. Some recent studies, I'll give you one example of gold label studies against Candida species, including some mutations. I won't go into great detail, just show you some of the highlights. Fungal systems have cell walls, and so there's typically an outer wall and an inner wall. Outer wall is dominated by mannan and the inner wall by glucans. And then you've got a more or less normal looking phospholipid bilayer membrane. And as we mentioned, this is where AmBisome does its business, but it has to get there. And the challenge has been to understand how this product gets through that cell wall and into that membrane surface. So we did a number of studies where we labeled AmBisome with gold and did TEM studies. This was done by Louise Walker, and it's been published in MBio. So I won't go through terrible detail here, but these are some examples where you can see the gold labeled AmBisome trafficking through both the outer and the inner layer and getting all the way down to the membrane. You can see that in the middle panel. Studies were done with two kinds of gold label, one where the label is internalized and one where it's tagged to the surface. And this was to confirm really that it's not about where the label is, it's really authentic behavior of the AmBisome particle itself. The free label itself will stick to that outer cell wall and not get anywhere near the membrane layer. With the AmBisome particle intact, it's trafficked through that cell wall and brought to the layer. And again, this is the interesting physics that we're trying to get our arms around. Different studies were done with different mutations. Some of the mutations are lacking in ergosterol, and those lead to lower clinical efficacy. Others have thicker walls, thinner walls. It's a whole series, whole library of mutations are being looked at to understand how different species might respond to the product. And again, two phenomena have emerged. One is if there is no ergosterol in the mutant, in this case, the drug itself will not traffic through that cell wall. It will stop at the outer wall. And if their drug itself is not on board and you're using a placebo AmBisome, it doesn't traffic. And so it tells you that amphotericin ergosterol binding not only works at a proximal level, but it tends to create what we called a porosity increase in the bilayer. I just focus on the upper graph here, which shows that if you have a wild type enzyme or a wild type candida and challenge it with various size dextrans, there's a certain relative porosity, and AmBisome tends to widen gaps in the cell wall in a manner that's not understood, but that's part of the study. These are some results of efficacy as it relates to those mutations. In some cases where there's little ergosterol or where there's thickening of the wall, you can start to see a response of the drug to these changes. Again, many of them, the wall is fully traversed, however. It just takes a bit longer. So it's really the rate of that transport. A short summary of all of this complexity I show you. For AmBisome, I go through both walls and come right to the membrane surface. An empty liposome stops at the through the first wall. The gold particles will go along with the AmBisome or the placebo accordingly. And if I have no ergosterol, the AmBisome gets stuck as if it were placebo. And so again, we continue to pursue these kinds of studies to better understand how the drug works and against which kinds of pathogens. So these are just a summary of things I mentioned before. And the physics really interesting is what is the relationship and nature of that amphotericin or ergosterol binding? Is it a transfer of the intact channel to the membrane surface, or is it something more complicated? I want to move on to some developing world use expansions. It was shown some time ago that cala-azar, which is the parasite that AmBisome is well suited to treat and contains ergosterol in the system. It's often fatal if left untreated in most cases. And in some cases, and depending on the region and some of the genetics of the parasite, it can be cured with one large single dose, or in a few cases, some more complicated dosing is necessary or a different agent. But it's highly effective against many forms of cala-azar. And this is because of the nature of the sterol skeleton element in the parasites. So we worked together. This is a case of, how can we get an effective use of either donated or relatively low-cost product provided to the non-government agencies that function in those regions, Bihar, South Sudan, and other areas where the product is endemic. So in 2011, a vial donation was made, and together with the Gates Foundation and a number of companies and other NGOs, the London Declaration was signed, and the target was to really wipe this parasite out by 2020. And this is where we are today, obviously, somewhat distracted by the other pathogen that's circulating the world. That donation was increased in 2016. And we began to hear good news from the ground that the product was doing really well, especially in areas like Bihar. And this really had to do with some focus on not only the drug itself and whether it works, but on logistics, distribution centers, protection of a drug that required a specific storage temperature, and elements of when to deal with the pathogen, in this case, a fly, companion animals, and trying to treat whole villages and whole population centers so that you don't have the drug or the disease continuing to spread. And this just tells you some of the challenges of actual administration of the drug. You're very seldom dealing with a sophisticated hospital setting. And so part of the challenge has been to focus on supplies, staff training, the temperature of the environment. The drug itself is not stable above 40 Celsius. And then dealing again with the disease vector. It doesn't help to cure someone and they get the illness again very shortly afterwards. And so did we accomplish the goal together? Well, I think we've seen a dramatic drop in cases globally. I think it's very exciting. Hasn't really made the news much, but we're down to a relatively low number of cases relative to just 9 years ago. The map shows places where we still have 1,000 cases active, but typically that map would have been 10 or 100 times that in years past. And so we're now focusing efforts on continuing to really get this down to zero if we can, but looking at other fungal infections that are endemic to this very similar regions, including HIV-related cryptococcal meningitis, which has a large number of patients, disseminated histoplasmosis, and other elements. And so there's another opportunity now to really go after these, perhaps in a similar way, although each of them have a different medical, sense of it. So what about new liposomes at Gilead? So Gilead has no other targeted liposome system in commerce, but we do continue to work on candidates. Some of them are RNA related, some of them are small molecule related. We did also try to purvey a technology of simple drug delivery, and the lipidic systems are actually quite useful as solvent systems. This is an example of a liposomal propofol that we made as really a demonstration vehicle. It's a lipid complex with the drug in the membrane, and typically the release of the drug is the same as you would have for a castor oil or cyclodextrin or other formulation system. So you're looking for pharmacokinetics of the same that is of the conventional care system for these systems, and really no uptake in liver or spleen, which are typical for lipid systems. And the other advantage it brings is you can give a bolus injection. You don't have to do dilution or infusion because the systems don't elicit these complement activation or other responses that are sometimes seen with the liposomal nanoparticles. This is an example of pharmacokinetics of a system in Chromophore versus a liposome. The one on the left is tacrolimus. The one on the right is etoposide. So again, these are demonstration systems. We also had quite a bit of experience in humans. We have a number of these products in phase 1 and 2. We've never seen carrier vehicle related toxicities from them. And we use them often to do placebo. This is a placebo of AmBisome that was used in a relatively recent clinical trial. So you can emulate the appearance of the nanosystem with these simpler delivery systems. The comment I make is, what do you do when you're pressed for time? So here comes COVID, and we'll talk a little bit about how this was chosen to be formulated, the drug Remdesivir. So Remdesivir, Gilead, of course, has a fairly lengthy and deep antiviral heritage, I would say. And it was developed in those days really as a generic coronavirus product focused actually on MERS. But it was part of a program against all of these viral families. When the 2014 outbreak came in the Congo of Ebola, the drug was produced the first time at scale as an injectable therapy. And at that time, it had been encouragingly effective in animal studies. It's a prodrug. The drug is on the upper right. And it ultimately needs to get into cells and be multiply phosphorylated to be active. And that first phosphate moiety is something that helps it to still get into cells and yet enable relatively rapid phosphorylation inside the cell. Somewhat late to the original outbreak, so there wasn't a whole lot of patient experience at the time that we first started, but there were two fairly famous cases. One was a young baby girl who had Ebola, where there had been, prior to Remdesivir, 100% lethality in those types of cases. She's still alive today. The other was a nurse who had returned to the UK after recovering from Ebola and had become reinfected by what became understood to be a fluid pool of virus, in her case, cerebrospinal fluid. It turned out also to be present in testicular fluid, to where the virus could hang around and reinfect certain individuals. So she was also treated, and it created quite a encouragement that a therapy could be had for Ebola. In the end, other drugs did better, and so the Gilead did not pursue an approval for Ebola, but we did gain significant patient experience. There was a number of conventional phase 1s that began, some eradication trials around that seminal fluid reservoir. And so at the time that COVID came, we actually had some filled vials in Southern California that were made available very quickly to one of the first patients that we treated, which was a case in our state of Washington. It was published right away in the New England Journal as an individual kind of anecdotal case. The dosing at the time was a 200 milligram loading dose, followed by daily 100 milligrams. At the time we started, which is hard to believe, it's only January or February of this year, we were looking at almost 10 days of dosing. It seems clear now that the extra 5 days don't help you. A number of clinical trials began, some Gilead-driven, some government-driven, and some NGO-driven. And so the challenge in supply became to supply them all with drug as we went forward quickly. Coming here today, we actually have a number of commercial approvals, including on the 22nd, the US approval of the drug. and we call it Veklury is the brand name of the product. Again, in some cases, the approvals are conditional. In other cases, they're what we would call a normal approval. The drug itself, as I mentioned, is insoluble and really not orally available for a number of reasons. We had a choice of trying a lipid system and of looking at cyclodextrins. In this case, the lipid system, they were both looked at and tried, and we have progress on both types of formulation, but the one that was ready to go extremely quickly was the cyclodextrin. And there are some advantages to systems where you simply add the drug and the carrier together, stir for a bit, and go. And the cyclodextrin system, although the specifics of the formulation had to be thoughtfully carried out, ultimately, for a new drug at the bench with relatively limited experience in formulating lipid systems, there is a certain barrier to entry that the lipid systems present. And the cyclodextrin system turned out to be very effective in this case, although it is expensive and it has its own challenges. The goal was to make as much as we could, as fast as we could at Gilead. There were both liquid and lyophilized presentations. Originally, the Republic of the Congo supplies were called back in and used to supply those initial named patients and also the beginnings of the clinical trials. Eventually, we had to say to the system we were going to get to a more general expanded use mode versus named patients, and then eventually, as quick as we could, get on to approval. Originally, we made the product exclusively in the Laverne, California facility of Gilead, but contracted many contract manufacturers to pick up production of not only the finished dosage form, but also the active ingredient. So a huge supply chain had to be built over the course of a very few months. And it was just amazing to see how fast that could come together. Most recently, there's still clinical data coming in, and Gilead's own website has continued to update progress, including fairly lengthy commentary by Gilead's medical teams. And so I encourage folks to look at the website. Actually, they have been quite transparent about the drug and its progress as it goes. The most recent study is a double-blind, placebo-controlled trial that was done by the NIAID in the United States and was published in the New England Journal of Medicine. And so it is this trial, among the others that have come through, that are the primary basis for the FDA approval on the 22nd. So that's what I wanted to share with you today. I'd be happy, obviously, to take questions. So I want to thank all of you. I want to thank our partner for AmBisome in Japan, which is Daiichi Sumitomo. I want to thank Professor Löffler and also Professor Hesse Baumhof for introducing me to ClinM. I was really looking forward to go. It would have been great. I want to thank our lipid suppliers. They never typically get acknowledgement in these systems. So I want to thank them. The academic centers that are continuing to study this product, the AmBisome product in particular, and the Gilead Access Program, which has been very effective at treating, not as effective as we would all like to be, but as effective in treating developing world disease, and our own technical teams at Gilead. Thank you very much.
Thank you so much, Gerard, for this wonderful talk. There are two questions. One is, I read it from Nathalie Minier. As it also has been observed by transmission electron microscopy, gold nanoparticles not attached to liposomes were also going to the inner layer, question mark.
No, so at least for the gold nanos, so we used a colloidal gold or nano, kind of nanoparticle evolved gold. We had two kinds. One was a free gold particle that was about 5 nanometers across. That did not go through. The other was tethered to a phospholipid, so that one was less surprising that it would stay exterior. But the small gold particle that would be on the inside of the AmBisome liposome was stuck at the outer wall surface in those studies. And even though AmBisome is much larger, it was able to traffic that cell wall, which was just, again, it's amazing to see, and it's an interesting phenomenon.
Thank you. The next question is from Michael Keller. What is, in your experience, the major reason that big pharma are still often shying off from nanomedicinal approaches to drug delivery?
So I think there's two basic issues. I think the first is an example of a repurposed drug or a drug that's out there. You have to look at the net increase in overall value of that drug. And in the case of AmBisome, it's somewhat unique because of the extreme toxicity of the free drug and how well it responds to entrapment. The other really successful drug, Calyx, for example, the doxorubicin drug, really was able to find a different place in the oncology regimens and turned out to be very effective. There are many other good drugs, but I think the idea is that if I have an old drug and I'm trying to up its delivery game a little bit, maybe the benefit isn't as good as it needs to be to justify the expense. The other is really just the barrier to entry technically. So a lot of companies, big companies especially, played with, and I'm of course thinking more in terms of liposomes, but a lot of complicated nanodelivery systems are just difficult to just try when you're doing that drug discovery phase and do that quick one for phase 1. And it takes a lot of expertise and energy to do that. And so we're just not seeing much of it in the bigger companies. Gilead does look at active ingredients with a whole range of delivery systems. But partly liposomes are in that mixture, partly because they brought in an old, one of the acquisitions was NeXstar. And that just doesn't exist everywhere.
The next question that comes is, no, it's a comment. Jon de Vlieger from Ligature is congratulating you and thanking you a great lot for this excellent talk.
Well, thank you very much.
Is double question. It was again Nathalie Minier and another person that wanted to ask you regarding Remdesivir. Natalie asks, how will their new Remdesivir formulations be administered?
Yeah. So the current
second question in the same sector.
Yeah.
Can you speak about successes and challenges with Remdesivir regarding COVID-19?
So I think to the first question, it is given IV. The patient has to be administered an IV infusion, and it's 200 milligrams on the first day, diluted in dextrose, and then 4 subsequent days of IV dosing. The big negative of that is they typically have to be in a hospital setting for us. And so the opportunity to dose patients who are positive or beginning to show symptoms, but are not in a hospital setting. In many cases, they're at home, they're still doing reasonably well. And so there's a choice to keep them, to not admit them to a hospital setting. Those patients typically won't get this drug. And so we don't know really how well they might do or might benefit from it. So there's that IV administration in this case does lead to that kind of basic limitation. Gilead is looking at inhaled routes of administration. That was announced. It's very different. We don't know how well that might work, but that's a potential that could be done outside of a hospital setting. One can do IV dosing outside of a hospital setting, but it is not simple. That's probably the biggest limitation. I think, in terms of success, we've been very fast at getting this drug from January or February start to today, when we have many millions of units produced. So it's been a great manufacturing success, and the clinical trials have enrolled very rapidly, although that's not so unusual given the prevalence of the disease. And really what we benefited from was having worked with the molecule extensively in about 5 years ago in the Ebola setting. So a lot of lessons had been learned, a lot of pharmacology understood, safety. So a lot of things had been, a lot of animal work had been done. So we had a large body of data when this thing started. So the progress this year has been amazing, but it benefited from that legacy.
Thank you very much. It is fantastic that Gilead as a company is going into challenges into effectively really into nanomedicine all the time, and since 30 years until now. And we think that your contribution is for us most valuable.
Thank you very much. Appreciate that very much.
We would love to have you next year or the year after or after COVID, let's say post-COVID, to host you in Basel in the best way. That is the only thing I can say.
That would be fantastic. Yeah, I really missed the opportunity, but I think you guys have done a great job in going ahead with this conference under the circumstances, and I think it's turned out to be a great conference.
Thank you so much. I would like to say to all people now that we are going at 2 a.m., 2 p.m., of course, continuing our session at 2 o'clock Swiss time, which is from now an hour and 15 minutes. There will be now a break. Thank you very much.